let's analyze the abstract of a life science paper written by Nobel Laureates. It is one of the best examples of how an abstract of a life science paper can be written. Let's read it and let's analyze what makes it such a beautiful abstract. When I say the word analyze, what do I mean? I will explain it to you as we go along today. When my students start reading a paper with me, my students will become usually pleasantly surprised when they see how I analyze the abstract, and they will start loving it. I will try simulating a part of how I would read a paper together with my students. I hope you will like it too. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologica. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto, learn the skills, enjoy academic life. Last episode. In the last episode, episode 45, we read the abstract of the Milestone Article of the 2023 Nobel Prize laureates in physiology or medicine. It was written by the laureates, Dr. Katalin Karikó and Dr. Drew Weissman, and it was published in 2005 in the journal Immunity. I'm sure you enjoyed reading the abstract yourself, because reading a part of the paper will give you much deeper experience than just hearing the news about a Nobel Prize winning work. We were able to read the abstract because we followed 2 substeps beforehand. In step 13-1, we checked the content of the Nobel Prize presentation video on YouTube. This video is important to us because we can get an overview of the laureate's work simply by watching it for only about 9 minutes. I cannot emphasize enough that watching this video is one of the most important steps for reading a milestone article written by the Nobel laureates. We have access to this beautiful presentation video, and this is the reason why we started our mini-series on reading by reading a milestone article written by Nobel laureates, not any other paper written by others. We would like to thank the Nobel Committee for preparing this video. Specifically, I would like to thank Dr. Sandberg, the Nobel Committee member, who presented the laureate's work so clearly. In step 13-2, we searched for keywords of the article in the title of the article. We found 4 keywords: the RNA, the toll-like receptors, the nucleoside modification, and the evolutionary origin. Keywords are important because they will help us identify the key concepts and the key statements in the article. Then, in step 13-3, we actually read the abstract several times. By reading the abstract, we could get the overview of the paper. If you understood the content of the abstract, that is great. But even if you did not understand the content of the abstract, that is okay. In the early phase of our reading mini-series, our goal is not to understand the content of this specific Milestone Article 1. Instead, our goal is to understand how an abstract is constructed and how a paper is constructed, so that you will be able to read any paper comfortably. This is because most of the life science papers are constructed in the same way, so you will experience the best benefit by going through our reading mini-series. Our milestone article will help you the most because it is one of the best examples of how a beautiful paper is constructed. If you have not listened to the last episode, episode 45, yet, please take a moment to check its content. It will help our analysis today. If you are new to my podcast and you have not read the abstract yet, please take a moment to read it before listening to today's episode. This is because at the end of today's episode, I would like you to read the abstract again so that you will be able to compare your understanding of the abstract before and after listening to today's episode. I would like you to feel the change, hopefully an improvement in your understanding. Today's episode. Today, we will analyze the abstract that we read in the last episode. And I would be happy to show you how I would read and interpret the abstract. We will go through one step today, and it has 3 sub-steps. Step 14, analyze the abstract structure of the article. Today's overall goal is to examine the content of the abstract so that we will be able to analyze the abstract structure. When I say to analyze the abstract, what do I mean by the word analyze? In our mini-series on reading, the word analyze means that we will read each sentence of the abstract or any section of the paper. We will examine the role that each sentence plays. We will assign each sentence to a structural component so that we will be able to understand the author's intention for each sentence. Let's get started with the first sub-step. Step 14-1. Review 8 structural components of the abstracts. In episode 39, we identified the 8 structural components in well-written abstracts, and we named them using our own words. By reviewing the structural components now, we will clearly see the structure when we read and analyze the abstract from now. Those 8 major structural components will usually occur in the following order: the background, the known, the unknown, the result overview, the methods, the results, the conclusions, and the implications. These components will play different roles in an abstract. Please let me repeat. Understanding the abstract structure is very important, as I will show you in the third sub-step today. If you have not listened to the episode 39, please take time to do so. Here, I will tell you 2 positive experiences I have had with the abstract structure. First, the experience by myself. After I realized these structural components, my reading skills improved drastically. With the 8 structural components in mind, I can understand the structure of any abstract I read in any life science field. Because I can understand the structure, I can understand the content better. Because I can understand the content better, the reading of abstracts becomes more fun. And because the reading of abstracts becomes more fun, the reading of the entire papers becomes more fun. Second, the experience by my trainees. I have been showing this concept of the abstract structure to my trainees. My trainees include, for example, the medical students, the graduate students, and the postdoctoral researchers. All of them told me, well, to be accurate, all the trainees that I talked with told me that it was their first time seeing a clear structure in the abstracts. And more importantly, they said that they were happily surprised to see how well they started understanding the abstract when they learned of its structure from me. So this is good news for you. Step 14-2. Give a sequential number to each sentence of the abstract. When we analyze the abstract, I will show you that each sentence of the abstract has a very special role. So we would like to identify each sentence in our discussion. We will do it by giving a sequential number to a sentence. The number is going to be the ID number of a sentence. In the abstract of our Milestone Article 1, there are 8 sentences. So we will give numbers 1 through 8. The first sentence will also be called the sentence 1. The second sentence will also be called the sentence 2, and so on. By this numbering process, we will start viewing the abstract not as a single block of paper overview, but as a section composed of individual sentences, with each sentence playing a special role in the abstract. This will be very helpful. Comment on copyright. Before we read the abstract together in the next substep, please let me give you a comment. It is about the copyright of the paper. Our Milestone Article 1 is an open access paper. This means that you and I can access the paper online without any account or without any signing in, and we can read it for free. But this does not mean that we can do anything about the paper. If we read the copyright statement of the paper is protected under the Elsevier user license. Its specific website indicates that there are strict rules about how to use the contents of the paper. I will list the URL in the episode description, together with the URL of the paper. Based on the copyright situation, I will not quote the author's sentences verbatim from the paper in today's episode and in the following episodes. I will paraphrase them. So when I refer to a sentence in my episodes, please read the corresponding sentence by yourself on your side. This is the end of the comment. Let's move on to the third and the last substep for today. Step 14-3, analyze the abstract. You and I will read each sentence of the abstract and analyze it. I will paraphrase each sentence for you, and I will add my comments. Our goal of this substep is to think about which of the 8 structural components each sentence is contributing to, and based on that, we will think about the author's intention for writing the sentence. Let's get started. There are 8 sentences. The first sentence says that when the nucleic acids, DNA or RNA, enter a mammalian body, they will be recognized as foreign, and they will trigger a so-called innate immune response within the body. The key player in this immune response is a group of proteins called the toll-like receptors that will detect such nucleic acids and start a cascade of molecular events that lead to immune responses. This sentence tells us the first structural component of the abstract, the background. The author's intention in this sentence is to start guiding us, the readers, so that we know what kind of topic to expect in this work. The sentence is a broad statement, but it quickly introduces the background information to us by telling us 3 keywords out of the 4 keywords we had selected before. The 3 introduced keywords are the RNA, the toll-like receptors, and the word mammalian, which is related to the keyword evolutionary origin. The second sentence says that some nucleic acids are exceptions. They are not recognized as foreign, and they will not trigger an immune response. One known example is the modified DNA, whose nucleoside, cytidine or cytidine, is methylated at a site called CpG motif. That is, cytidine is modified by receiving a methyl group. This sentence tells us the second structural component of the abstract, the known. The author's intention in this sentence is to tell us what is known in the field. Of course, there are many known things in any field. This known component is specifically related to the target of this research. The modified DNA somehow avoids triggering an immune response. The most important keyword appeared in this second sentence. That is the word modified, and it includes a specific modification, methylated. It is interesting to see that the keyword modified was introduced, but it was not related to the RNA, which is the target of research in this paper yet. It was related to DNA. The concept of DNA modification was introduced in the second sentence before the concept of RNA modification would be introduced in the next sentence. So the sentence 2 forms a bridge between the sentence 1 that talks about the DNA and RNA triggering innate immune responses, and the sentence 3 that talks about the RNA modification. For me, this second sentence was the most important sentence in the abstract. This sentence is closely linked with the next sentence so that we can understand the next sentence very easily and logically. This second sentence is the most important sentence because it bridges sentences 1 and 3 and serves as a logical structure, as we just discussed, but also because it gives us the idea about how this published work came about. This is a beautiful sentence. Then the third sentence says that, similar to DNAs, as mentioned in the previous sentence, some RNAs have modified nucleosides, but it has not been examined whether those modified nucleosides in RNA can avoid triggering an immune response or not. This sentence tells us the third structural component of the abstract, the unknown. The author's intention in this sentence is to tell us a question. Of course, there will be many unknown things in any field, but this unknown component is specifically related to the target of this research. The answer to this unknown is what the authors did not know, and this is what the authors wanted to examine in this paper. So we can expect that the sentences that follow will talk about what the authors actually found in their work. This sentence is a very clear statement. It is not an open-ended question. It is a question that can be answered with a simple yes or no in the simplest case, or answered with variable results in a complex case. Any result would be meaningful to the field. The following 3 sentences, the sentences 4 through 6, describe another structural component of the abstract, the results. The 4th sentence says 2 things. Both were based on model non-immune human cells in culture called the HEK293 cells. They were genetically engineered so that special toll-like receptors could be expressed in those cells. Those cells allow the researchers to easily control the subtypes of the toll-like receptors to be expressed in the cells. One of the 2 findings was that when the RNA contained unmodified nucleosides, that is, when the RNA was unmodified, the RNA triggered an immune response. And this response was mediated by specific toll-like receptors when the unmodified RNA interacted with them. Another finding was that in contrast, when the RNA contained modified nucleosides, that is, when the RNA was modified, the same immune response was not triggered. This is the result number 1. The author's intention in this sentence is to tell us one set of results. This is an outstanding finding. The sentence already answered the question, that is the unknown, of this work. The question was, do modified RNAs avoid triggering an immune response? The answer is yes. The sentence included 5 modified nucleosides that the authors tested. The last one was pseudouridine. Please remember that this modified nucleoside was emphasized in the Nobel Prize presentation video on YouTube. Please also note that this result was based on the model cells. The 5th sentence says that the authors extended the research from the model cells to more physiological cells, that is, the cells responsible for innate immunity in the body, called the dendritic cells of humans. The authors found similar phenomena to those found in the model cells. When the RNA contained unmodified nucleosides, the RNA did trigger an immune response. In contrast, when the RNA contained modified nucleosides, the same immune response was not triggered, or it was reduced greatly. This is the result number 2. The author's intention in this sentence is to tell us the second set of results. This result number 2 was based on the dendritic cells. In the result number 1, the authors used the model cells. The advantage of the model cells is that genetic engineering is easy in those cells, so that the authors could identify the specific toll-like receptors involved in the RNA-induced responses. But the disadvantage of the model cells is that they are not native immune cells, so it was not clear whether the results found with the model cells can be obtained similarly in native immune cells. In the result number 2, this disadvantage of the model cells was overcome by using native immune cells, that is, the dendritic cells of humans, and specifically multiple types of dendritic cells. So the result number 2 is a powerful statement and extends the result number 1 in a strong way. The 6th sentence is about the evolutionary origins of RNAs. The sentence says 2 things, both based on human cells that express toll-like receptors, that is, the dendritic cells and the model cells. One finding was that when the RNA was derived from bacteria or human mitochondria, both of which were rich with unmodified nucleosides, the RNA triggered an immune response. Another finding was that, in contrast, when the total RNA was derived from human cells, which were rich with modified nucleosides, the same immune response was not triggered. This is the result number 3. The author's intention in this sentence is to tell us the third set of results. The result number 3 integrates the mechanistic views obtained in the results 1 and 2 into a more biological context. Human mitochondria are thought to have evolved from ancient bacteria. Both bacterial RNA and mitochondrial RNA were rich with unmodified nucleosides, and they effectively triggered immune responses. In contrast, the human total RNA is rich with modified nucleosides because, although it includes mitochondrial RNA, it is dominated by ribosomal RNA that is rich with modified nucleosides. By the way, the introduction later says that the ribosomal RNA occupies about 80% of the cellular RNA. And this human total RNA did not trigger immune responses. So the result number 3 introduces an evolutionary viewpoint. The 7th sentence says that the results obtained in this work show that when the RNA contained modified nucleosides, the immune response by the dendritic cells was greatly reduced. This sentence tells us another structural component of the abstract, the conclusions. The author's intention in this sentence is to summarize the findings, the results number 1 through 3 presented so far. By summarizing all the findings, the conclusion statement gives a clear answer to the unknown stated earlier. Do modified RNAs avoid triggering an immune response? And the answer is clear yes. In the 8th and the last sentence, the authors proposed their idea. In the result number 3, the authors showed bacterial RNA and mitochondrial RNA triggered immune responses. In their previous work, the authors also showed that the RNA from necrotic tissues triggered immune responses too. Necrosis is a cell death due to abnormal damages. And during necrosis, the RNA rich with the least modified mitochondrial RNA is thought to be released. So it is possible that modified nucleosides are important regulators of how RNAs are recognized by the innate immune system in the body. This sentence tells us the last structural component of the abstract, the implications. The author's intention in this sentence is to describe what is implied by the findings. The authors placed their findings in the work in a broader perspective. They provided the idea that the degree of RNA modification could systematically explain the immunogenicity of RNA under various conditions, including the evolutionary origins and necrotic changes. So, in this abstract, 6 of the 8 structural components of the abstract were used. The structural components used were the background, the known, the unknown, the results, the conclusions, and the implications. They were laid out beautifully. The abstract of our Milestone Article 1 followed the nature guidelines that we talked about before. And we saw that each sentence contributed to the structural components of the abstract. The best component that I found to be the most helpful was the known component. What we did today. I know that we talked about the very rich content of our abstract. Let's stop here today. Today, we started step 14, analyze the abstract structure of the article. Specifically, we covered its 3 substeps. Step 14-1, review 8 structural components of the abstracts. Step 14-2, give a sequential number to each sentence of the abstract. And step 14-3, analyze the abstract. In the following episodes, we will keep analyzing the abstract. We will go even deeper. Your action. After you finish listening to this episode, please read the original sentence of the abstract again. Do you now see that each sentence is actively playing its own role? Do you see the author's intention for each sentence? Do you feel that you have understood the structure of this abstract completely? If you feel that way, that will make my day. I feel happy for you. It would be great if you could tell me how you felt. Was today's episode helpful for you? Or did it have no effect on your reading? Either way, please let me know. I would like to hear from you. Your feedback is important to you and me because it will help improve the later episodes for you. My email address is ideas@synaptologica.com. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.